Hello. Today we're going to talk about ancient DNA, and we're going to ask the question, how far can we see in the past? And I'm going to answer that question by examining a very short article by a creation scientist that misrepresents or misreads or possibly doesn't read the scientific literature on ancient DNA. Now, just to remind you, my name is Joel Duff. I'm the author of naturalhistorian.com. I look at the intersection of science and faith, and I'm very interested in DNA. I study genetics and in a variety of different systems. I've actually done a little bit with ancient DNA myself. I've extracted plant specimens that are up to 80 years old, so we're not talking about millions of years or anything like that, but even 80-year-old DNA is pretty challenging. And so I've learned something about the challenges of working with ancient DNA. I teach genomics, and I have a section in that class in which we discuss and read a couple papers about ancient DNA in order to understand something more about how we can identify problems with the structure of DNA and its decay over time. So what inspired me to put together this short presentation? Well, a few days ago I saw a tweet by David Rives, who's a creation apologist who promotes a variety of different of his own ministries that I'm going to call a kind of a fringe creationist organization. And he was promoting a particular article which was just published on one of his blog or websites, the Creation Club. And on that, and he was claiming that this article had proof that the world must be very young. That scientists clearly have shown the world is young because they've sequenced samples that they think are from super old samples of 100 million year old weevils. And since we know the DNA can't last that long, if they really have found DNA in this amber, which they think is very old, that DNA is actually proof that the world is young because the DNA can't have survived very long, so that amber must be very young, probably produced in the global flood just 4,000 years ago. All right, so that caught my eye. I went and I looked at the article. And the article consists of about 3 paragraphs, and I'm going to show you all 3 paragraphs, and we're going to use this article to discuss a little bit about the scientific process, how science progresses over time in our understanding of topics such as ancient DNA, and how many creation scientists seem to be living in the past and haven't progressed with it. Right? They haven't really followed the literature in order to understand the literature of ancient DNA, and therefore many of the claims they're making in the present about past studies are very deceptive, or if not deceptive, at least ignorant. And I think in this case there might be just ignorance, but ignorance nonetheless is leading other people to be ignorant because they're using these to make very strong claims about how this is proof of a young Earth, and yet not so much proof, as we are going to see. So I do want to say something about the particular site that this particular article was found on. I think it's important to know your source, and I think it's a little bit informative about sort of the culture of creationism. How there's sort of different tiers of creationism, right? You have your Answers in Genesis, your Institute for Creation Research, your Creation Ministries International. Those are your 3 main organizations that are devoted to promoting creation science, the use of science to try to prove that the world is young and is compatible with what they believe is their what is their interpretation of the scriptures as supporting a young Earth. So, but this particular, those would be the primary organizations. Well, surrounding those organizations, there's a whole lot of other, I'll say creationist wannabes who would like to be big organizations and would like to do the big business of creationism. One of these is David Rives, who's an independent creationist, but he's more than a creationist. He's an overall apologist because he has a ministry that involves a whole bunch of different branches, but he has a special fondness for creationism, as we'll see. And one of his groups is called the Creation Club, and that's where this particular article is found. So the Creation Club is one of David Rives' ministries. David Rives seems to be an independent apologist, has a great affinity to creation science. He is an amateur astronomer, and that's sort of where he got his start, was talking about astronomy, and that's mostly what he writes about, but he's expanded to sort of talk about all the different sciences. I'm very unclear about what his educational experience is, if he has any at all, because all the different profiles of him simply talk about his expertise in these areas being derived from his experiences in these areas. So I guess he's read a lot or actually been involved in doing some kind of dinosaur dig or something like that. And so he's claimed experience from that, but not at all from his educational experience. Not that there's anything necessarily wrong with that, but we'll see that he's a self-made person, a self-made promoter, and promotion is what he does. I mean, he has all these different web pages and sites. He has a store for selling material, the Creation Club. He has little blurbs that are on Christian radio, so he comes on and he gives these little talks. He takes you on safaris that are creation organized. He's working on this big video or movie about the Grand Canyon. He's written his own music that he'll sell to you, and so forth. You get the idea. This is a one-man show, although there's multiple other people involved in producing this particular site. But one of his, like I said, he's a sort of a self-made creationist. He's sort of wants to be the replacement for Ken Ham someday, I'm sure. And so he has this site, the Creation Club, and on that he cultivates a variety of different articles that pop up every day. And most of these are done by authors who obviously have looked at the creation materials produced by Answers in Genesis, an Institute for Creation Research, and they've sort of regurgitated that material and turned it into short articles, bite-sized articles for places like this. And the authors that mostly write for and are promoted on this particular site are ones that I've never heard of, and I'm very familiar with creation science overall, having studied it and worked with it and know people in it from the last 20 years. So usually these are folks that, again, have probably learned everything they know about science from creation scientists, and they're on the fringe of creation science, right? And in a way, this is what makes these sites particularly, in my mind, damaging, or dangerous, because the information that's provided here is highly suspect. I mean, the information that's provided by Institute for Creation Science and Answers in Genesis, they at least have editors who have a chance of finding the errors that are involved. They can at least ask each other whether what they're saying makes sense. And although they have a particular bias that makes them view things many times, I think, incorrectly, they're more likely to be at least somewhat on the right track with some of the things they say, at least in how they reference articles and understand the literature. But then when others read their literature, don't necessarily understand it, mix it with their misunderstanding of science, and then write it again, and then are even more dogmatic, they tend to take whatever errors were already there in the Answers in Genesis material and then accentuate those errors and make them even more dramatic. And this is going to be an example of that. So here's the article. A bug entombed in amber still had typical DNA. And this word typical is important, as we're going to see in a few minutes. So it's by Bob Enyart. And again, I didn't know who Bob Enyart was. So first thing I asked myself is, who is Bob Enyart, and on what grounds or what authority, what expertise does he have to write on this particular subject? And I found out that he is, and this is the little bio that's there, and I also did some additional searching, but he's the co-host of something called Real Science Radio. So that's something you would hear on some Christian radio stations. Along with Fred Williams, who I could find nothing about, but I'm going to presume he knows something about science. Maybe he's a high school science teacher or something like that. I hope he knows something about science, because it's pretty apparent that Bob Enyart doesn't have any background in science. He's a talk show, a right-wing talk show radio host, and he's a pastor of the Denver Bible Church. But the two of them get together, talk about science, debunk evolution, show the evidence for the Creator God from biology, geology, astronomy, physics, right? They make their own discoveries and inventions and breakthrough reports. I've really not gotten to see what exactly that is supposed to mean, but from the articles I have read from him, it's hard to imagine what kind of breakthroughs they're making. Nonetheless, this is the author of this particular article. And like I said, I think everything he's learned about science probably comes from reading creation science material from Answers in Genesis and the Creation Research Institute and other areas, or Institute for Creation Research. So here's the article. 3 paragraphs. Here's the first 2. He tells us that scientists have extracted DNA from 130 million year old weevil. And where do we learn this from? Well, nearly 30 years ago in the journal Nature. So this is the report he's using. This is the evidence he's using in his paper, is a 30-year-old report from the journal Nature. Now, Nature is one of the top 2 science journals in the world, rated journals in the world. So it is a very prestigious journal. And in that journal, they had reported that there had been the successful isolation of a 130-year-old weevil DNA from amber. Now, this should conjure up images of Jurassic Park, right? And this is really where the inspiration came from by Michael Crichton for Jurassic Park. The idea that maybe amber, in which you see these insects that are preserved in there in wonderful morphological detail, the fact that in that amber, if the morphology could be so well preserved, and the amber just looks like a sealed package, right? Maybe there could be the DNA molecules inside of those insects. And so Jurassic Park was all about the idea that if you drilled in there, you pulled out some material from one of these insects, you could extract the DNA from that insect, in this case a mosquito that had drunk the blood of a dinosaur. And so you'd have the dino blood, right? Or the dino DNA from that blood. And then you could take that DNA and you could fuse it with a modern reptile, and then you could create basically a dinosaur from that. All right? Great science fiction. And this almost makes it sound like science fact, right? Because if you can really extract DNA from a 130 million year old weevil, maybe we really could extract DNA from a dinosaur bone. And in fact, I'm going to suggest that creation scientists should actually believe it's possible, because if the world were deluged 4,350 years ago and all the dinosaur bones came to be formed just 4,000 years ago, I would expect, based on what we know about DNA preservation, that we should find lots of dinosaur DNA. We should be able to reconstruct the entire genome of a dinosaur if it had been preserved only 4,300 years ago. All right, we'll come back to that. All right, so he recounts correctly that they had extracted DNA. They used this process called PCR to amplify a segment of the genome of the organism they had extracted. They sequenced the 18S gene, a common gene, and then what they did was they compared that sequence to modern sequences of a variety of different insects, and through that comparison they were able to tell that this sequence was most similar to a weevil, which is why they said that they had sequenced a weevil. It was through the DNA comparisons. Great. That all sounds really fascinating, really interesting. So what's his main point, though? So why bring this up? What does this have to do with proving that the world must be young and cannot be ancient? He goes on to say that, okay, wow, I mean, obviously they've overstepped their bounds. DNA can't last that long. You know, rather than the existence of endogenous DNA, endogenous means DNA that actually came from an organism that was inside of that amber. And that amber pretty clearly came from a rock segment, right, from rocks that were dated to be 130 million years old. All right, so yes, okay. Now, he would disagree. Those rocks aren't that old. They're really only 4,000 years old. But he said, if you geologists really believe that these rocks are 130 million years old, and that amber is 130 million years old, then the DNA you just extracted is 130 million years old. But we know that DNA, it's just crazy to think that DNA could survive that long. It would have to decay. It's a sensitive molecule. It couldn't have survived 130 million years old. So if this really is weevil DNA sequence, and he seems to believe it is, right? He's convinced. This article is published in Nature, and by real scientists who extracted this DNA, and they are saying that it is weevil DNA, and he believes it. And so he said, hey, if that's true, it can't be true, right? So one thing, if it really is weevil DNA, which he believes it is, then the other thing can't be true. It can't be 130 million years old. If it is 130 million year old, then the DNA can't actually be from a weevil because that DNA couldn't survive that long. Now he's not accepting that proposal. He's willing to accept that it actually is weevil DNA from the actual specimen itself. In which case he thinks that proves that the world is young, because it would have had to have been preserved recently for that DNA to survive. Right? And so he's accusing the scientist of not daring to consider out loud the possibility that this is actually evidence that they found that the world is young, because they might risk losing their careers. Now, as we'll see later, this particular scientist hasn't had exactly an illustrious career after this, because he made a lot of other claims of finding ancient DNA, and all of them have essentially been debunked. No serious scientist today actually believes that there was weevil DNA from 130 million years found inside of this amber, but we'll get to that. But if you want to read more, I've written about ancient DNA several times on my blog. In this case, here's an example. Where's the DNA? Young Earth Creationism and the Search for Ancient DNA, in which I talk about the problems and challenges of working with ancient DNA, and how young Earth creationists should consider the challenges to young Earth creationism from the actual existence of any ancient DNA at all. Let's look at the original paper. Let's talk about that original Nature paper, because I suggested that there is an alternative explanation to what Bob is saying here, and that is you have to consider the other hypothesis. Maybe it actually isn't 130 million year old weevil DNA that they discovered. Right? Maybe it's a contamination, and that's what everyone believes it is. So here's the original article: Amplification and Sequencing of DNA from a 120 to 135 million year old weevil. And so it talks about going into this piece of amber, extracting pieces of material from it, and then extracting DNA, and then amplifying DNA, and then sequencing it. And then after they sequenced it, they lined that DNA up with other known sequences from modern organisms, a variety of different kinds of insects, because they knew they were looking at some kind of insect in the amber. And lo and behold, there was this weevil, which was most similar to the fossil weevil. So what they've done here is they've lined the sequence up, and they use a computer to analyze the sequences and just ask the simple question, which two are most similar to each other? And then it draws a phylogeny, or a depiction of the relationships based on their sequences. And what they discovered was their fossil sequence was very similar to a weevil, and therefore they concluded that, hey, it's not exactly the same as a weevil today, it's different, but it's similar enough that it probably is a weevil, but different enough that it could have come from a long time ago, and therefore had, there's been mutations over 100 million years. It seemed like a reasonable hypothesis, a reasonable conclusion, that it looked like their sequence was not exactly the same as anything alive today, and so therefore could be an ancient DNA sequence. This is one of the reasons this was published. You might ask, why did Nature publish this if it's so wrong? If no one believes that today? You have to remember that 30 years ago, this was really at the beginning stages of our being able to sequence anything, much less ancient DNA samples. There have been a few examples. As soon as you sequence modern things, of course somebody's going to be like, hey, how far can we push this? I can sequence something that I preserved from a year ago, 10 years ago, 100 years ago. I can get hair from a specimen from the museum. I've gotten museum samples of plants, and I've extracted DNA from those. And you can ask, how far back can I go? 1,000-year sample. What about mummies? All right, from Egypt. I know approximately how old they are. Can I get DNA from those? Yes, you can. And so there's always going to be somebody who's going to push the envelope, right? And it's like, what about ancient bones? Can I get DNA from those? Well, a couple people really tried to push it far, right? This amber thing, and this is not the only amber example. It was like, I can go into this amber, get DNA from those specimens. I could be looking at samples that are 20 million years old or 100 million years old. And this was one of the most dramatic examples, the oldest at the time when it was published example. So, at the time, nobody really understood how difficult it really would be. There was an expectation by many scientists that it really wasn't probably going to be possible to extract something from 100 million years old, because biochemically you would expect it to be difficult for DNA to survive that long. in its normal configuration. And so it would have been surprising to find this result. Nonetheless, there wasn't, the evidence presented in this paper is reasonable given the technology of the day. Now, even after it was published, there was a lot of skeptics, right away. It was like, all right, that's interesting. I can't prove you're wrong, but there's good reasons to be skeptical. And now we know there's a lot of reasons to be skeptical, and we have really good evidence that this particular result is completely bogus, that it's actually contamination of a modern weevil. But I do understand. I understand how this could have come about at the time. Nature is all about publishing sexy, cutting-edge stuff, and the write-up is decent, and so they got it published. But I can tell you, today you could not publish this work in this particular state, and I'll give you a couple reasons why. But let me move on to show you a couple other more recent articles. So this is in 1997, the Proceedings of the Royal Society, so another prestigious journal. These authors, I picked this out just because of the summary here. Apparently, ancient DNA has been reported from amber-preserved insects many millions of years old. Rigorous attempts to reproduce these DNA sequences from amber and copal-preserved bees and flies have failed to detect any authentic ancient DNA. Lack of reproducibility suggests that DNA does not survive over millions of years, even in amber, the most promising of fossil environments. That's the conclusion they came to in 1997. Many scientists tried to replicate the results of these suggestions that ancient samples could be amplified. And they either completely failed to get any DNA at all, or they discovered for themselves that what they were sequencing were contaminations in the environment. So how might contamination have happened back then? Well, I'm going to tell you that they didn't follow the procedures that you would have to follow today if you were sequencing or trying to amplify ancient DNA. So if I were to want to sequence something that was 500,000 years old, said to be 500,000 years old, I would take that sample and I would send it to a special lab, kind of like a carbon-14 analysis. I would send it to a special lab that specializes in doing ancient DNA extractions. And what they do is they have ultra-clean labs. I would send it to a lab where maybe they've never done any sample of that, anything related to what my interest is, or my sample is. That way, there's less likely to be contaminated. I might also send two samples to two separate labs, so I can get independent verification of the sequences that I get. Right? What they did was they did this in the same lab they do all their other work in. And if you're working in entomology, you're working on insects, and your interest is in insects, you have a lot of insects in your lab, and maybe you're extracting DNA from other insects. So then you pick up your pipetter, which you've just used to extract, your modern sample, and then you go over and maybe you think you've cleaned it out, but in the air, in the pipetter, in your solution somewhere, you've got modern DNA. It's all over the place. And as you do your extraction, there really isn't anything at all inside of that amber. But as you do the extraction, you basically add modern contamination to it. And PCR is a super sensitive process where it can pick up on a single molecule. One single copy of a contaminant can be picked up by PCR, amplified to the point where you can then study it, sequence it, and get the product. And you think that you have amplified something from 100 million years ago when you just amplified something that was sitting on your lab bench the day before. This happened all the time in the 1990s. Before we developed, scientists started recognizing, these are not real results. We need to have protocols in place to prevent this kind of thing. And you need to, if you're going to publish in the future, you need to show you went through these protocols. You need to show all these steps you've gone to show that you're not actually sequencing modern contamination. There's another problem with this particular paper. I would have loved to have checked this for you, but I couldn't, and I attempted to. All of these samples here had gen baking sessions. Now, what that is it's a code. It's a letter designation for the accession for the national database, or really a worldwide database of DNA sequences. So I could go in and I could find all the exact same sequences they used, and I could show them to you. The problem is they didn't actually include the GenBank sequence for the fossil weevil sequence that they developed. Again, today I couldn't publish a paper without actually submitting my sample that's part of my study to the worldwide database, getting the accession number, and I have to put it in the paper. The paper would not be published without that. Because if I publish a paper with an ancient DNA sample or any DNA sequence, it has to be available to every scientist in the world. So as soon as my paper is published, they could go get that sequence, they could do the exact same analysis I just did, and they could test my results, right? They could also use my sequences for any other study that they wanted to use it for. That didn't happen in this case. But here's the interesting thing. Look how many samples they used. These were really all the samples they had available to them in 1993. There weren't a whole lot of insect DNA sequences out there because it was very expensive and difficult to do DNA sequencing even of modern samples, much less ancient samples. Today, if you went to GenBank and you asked for weevil sequence, you'd be hit with hundreds of thousands of DNA sequences and probably tens of thousands of sequences of the exact same portion of sequence that they used in this study. If they had had those available, they never would have published this paper because they would have found out just like that they sequenced a modern contaminant. Why? Because they would have put hundreds of weevil samples into their analysis, and this fossil weevil probably would have ended up being 99.99% the same as one particular species alive today. And they would have known that you can't have a sequence that's 100 million years old be exactly the same as a sequence today. You cannot have that much time pass without mutations happening and changing the sequence. So today, it's pretty easy to identify that kind of contamination because your fossil sequence should be somewhat different than your modern sample. But there are other problems as well. And there's another way to tell you're looking at ancient DNA. And this is something that wouldn't have been available to the authors in 1993. And this really hasn't been developed until this paper is in 2014. But I would say 2010 to present, we've become much more conscious of how to look at sequences and tell how much DNA damage they've experienced over time. So this is called postmortem decay in DNA, and we have followed postmortem decay by actually following DNA samples that have been preserved and held on to and then restudied at a later time to look at how biochemically does DNA degrade over time. So in this example, now they're showing they're giving it a postmortem decay score. You can just think of that as the amount of decay. And if this would be a sample that has virtually no decay. And in this case, what decay is and I can't go through all the biochemistry, but it's individual places in the DNA molecule that are more prone to having biochemical changes. So not every position changes at the same rate. Certain base pairs, A, T, C, and G, will change more frequently than others. And so some will be converted from one base pair into another through a biochemical process that happens naturally over time. Really by chance processes, and decay that molecule such that it changes its molecular code over time. So if you're looking at a modern sample, right, you don't see any code, you don't see any decay. If you're looking at a 100-year-old, in this case Australian hare, you start to see a small amount of change in the DNA structure, the sequence that's there. And this 5,000-year-old human sample has all kinds of different changes. And this 38,000-year-old Neanderthal, now in the creationist timeline, of course there's nothing over 6,000 years, but we'd say the Neanderthals are at least 4,000 years old, right after the flood or something. So they're old in a creation model. They all show distinctive patterns of biochemical decay in their DNAs. So going back to our 30-year-old Nature paper, when they got their sequence, they didn't know that there were distinctive patterns of decay that could allow you to look at a modern sample versus an old sample. And just by looking at the patterns of usage of ACs, Ts, and Gs, you can identify samples that are older than others. And And so this is, for example, how do you, when they extract DNA from a femur from a Neanderthal, there's always modern contaminants involved in that process. So how do they separate the modern human contaminant from the Neanderthal sequence? One of the ways, there's multiple ways they can do that, but one of the ways they can do that is they can look at the final sequences they get, and they sequence hundreds of thousands of fragments. They can look at each fragment, and they can see that some fragments have a skewed pattern of A, T, C's, and G's that would not be expected given the particular genes they're looking at. Whereas in the modern human sequence, they get the expected profile of the distribution or ratios of the A, T, C's, and G's. And so they can identify particular sequences as having experienced this post-mortem decay, whereas others haven't. And so that's one indication of old versus new. So again, in 1993, no idea, right? You just get the sequence and, well, that's the sequence. You don't realize that there are processes that have skewed or changed the actual code of the sequence you're looking at, so now you're not looking at the original code of that sample. So if you got a sequence that you think is 100 million years old, and it comes out and it has no post-mortem decay, and it has an exact same sequence as a modern species, there's no way that it's a 100 million year old sequence. And that would have been true for all those samples from the 1990s, which claim to be 10 or 20 or 50 or 100 million years old. All of them show no evidence of this pattern of post-mortem decay, and therefore are modern contaminants. Now, the point of all this, of going into all that, is to show that DNA science has progressed a lot over the last 20 years. We can identify things that we just didn't understand in the past. Now, this author comes along and he's pointing to a 30-year-old article as proof that the world is young. But he shows no evidence that he has any idea what the science of ancient DNA is. He just trusts that, well, this is in the literature, so therefore it must be true, right? It's in Nature, a perfectly good journal, and therefore, scientists believe this. They think that all of us must be going around thinking there must be 100 million year old weevil DNA that really exists in the world, when really no modern scientist, if they know anything about ancient DNA, is going to accept those results. It's going to require a lot. You would have to get a different kind of result to have any acceptance of that. And this is why no one really accepts that there is good, beyond maybe a couple million years, and most people would say under 1 million years, there's a lot of ancient DNA that's available to us. But once you get over 1 million years, it's only going to be in very exceptional conditions as you're going to have enough preservation to slow that post-mortem decay down to the point where maybe we can get small fragments of DNA. Now, I need to add one other thing. And I need to acknowledge that not all young Earth creationists are completely oblivious to the literature of ancient DNA and mangle the results of this and misunderstand the history of science in this particular field. Answers in Genesis, here's an article from 2012 by Elizabeth Mitchell. She talks about ancient DNA decay, because there was a paper that she reviewed about looking at how fast this decay process occurs, and it concludes that really most DNA decays within the first 10,000 years. Some DNA can survive a lot longer, but most of it decays fairly rapidly. And so she even acknowledges that these older reports of millions of years old have been dismissed by scientists for appropriate reasons. But that again illustrates that Bob who's writing this article about ancient DNA, using this article about ancient DNA, doesn't even know the young Earth creationist literature well enough to understand that they have progressed in their thinking as well, because they did use that Nature article in the past, and he's probably just picked up on that and is repeating it. He didn't do enough research to find out that those who he's following have changed their minds, and he hasn't progressed in his own thinking to understand that. And that's what happens with these fringe organizations. They're so excited about whatever they see that they can glom onto that they feel supports their position. They don't do any further research to find out whether even their own viewpoint has been turned over by others in their own field. So I do want to give credit to Elizabeth Mitchell and others for recognizing that. Now, I do think that they have their own problem. Yeah, I think that young Earth creationists have an issue with ancient DNA. It's not all... they'll say that, look, DNA can't last very long, right? So therefore, any reports of 10,000 or 100,000 or even 1 million year old DNA should be a problem. I don't really think that's a problem. I don't have any problem with DNA surviving that long. I don't see any evidence that it can't survive that long under certain conditions. I would flip it around, and I would say that young Earth creationists have a little bit of a problem. If DNA can readily last for 10,000 years under fairly good preservation conditions, and the world is only 6,000 years old, then many fossils should have ancient DNA in them. In fact, I would say that young Earth creation scientists should predict that we should be able to sequence dinosaurs. Why not? They were all preserved 4,350 years ago in a huge flood, which was a pretty good preservation event. And some of them we should be able to, we actually have found dinosaur bones that have intact bone marrow type material in them, right? We should be able to extract DNA from them. They should predict that we should be able to get DNA from those and extract them. Whereas evolutionary biologists would say, or geologists would say, look, those bones are 65 million years old. And now we have pretty good evidence that DNA really doesn't survive that long. And therefore, we wouldn't predict that we'd find DNA in dinosaur bones. The fact that we haven't found DNA in dinosaur bones, I think, is a problem for young Earth creationists. I think we ought to be able to be able to get DNA from virtually every organism that has been on Earth, because DNA preserves well enough that we should be able to find at least small fragments of remnants of most organisms that have survived and been preserved in the fossil record. But we don't find that. We find it's very limited to the very, very, very top of the fossil record. We sequenced an entire mammoth, all right, from mammoth tusks and teeth. And those mammoths may have lived 10,000 years ago or 50,000 years ago. Or in the young Earth creationist timeline, they lived 4,000 years ago, like right after the flood. If we can sequence an entire mammoth and get pretty high quality DNA from them, and it's 4,000 years old, why can't we get high quality DNA from a 4,300 year old dinosaur bone? Now, why is it all of a sudden that you get to 200 years older and all of a sudden the DNA just seems to disappear? This is a problem for young Earth creationists that I don't think that they've really grappled with or come up with a model to explain why they're not getting the results that their own models essentially would predict. All right, thanks for tuning in, and we'll talk to you later. Bye-bye.